Yeah, so yesterday we were talking about olfaction, and today we'll switch things around and talk about navigation, the other behavior that this wolf in my illustration seems to be doing. Uh, so, as usual, before I start, I put my acknowledgement slides because this talk I have actually given this talk or some version of. I should never say I've given this talk before, right? I've given some version of this talk before, and I've never gotten to the end of it. So therefore, the acknowledgement slide. So. The work that I'll be talking about was initially started off with a graduate student in my lab, Arun Nehru, and much of the work that I'll be presenting is done by another graduate student, Inayat Sheikh, who's in the lab now. He if we get time, I might cover some of the work that Divyansh, who's an undergrad, who was an undergrad in the lab, and Srirang did while over the last year or so. And I'd also like to thank people from Suhita's lab and our funding agencies for generously funding this work. So, what we're going to look at today is essentially thinking about how we represent space in our brains. All right, and an interesting a sort of set of experiments that people looked at is due to Edward Tolman. All right. Edward Tolman, by the way, his brother is Richard Tolman, who's the physicist, did several things in statistical mechanics, and Richard Tolman is a eminent psychologist. He worked, I think, in Berkeley for the longest time, and he was known for doing a number of experiments of rats going through mazes, trying to figure out how do they represent space? Can we do a lot of these experiments to get a sense of what's represented in the brain? And here's a really famous example of one of the mazes that Tolman built. So this is what's known as a starburst maze for obvious reasons. So what you have out here is, so you're looking at this apparatus from the top down. So you have a circular chamber out here, and a rat enters this particular circular chamber through this path out here. Now there is some reward or food that's at this particular location G. All right. So what the rat did over multiple trials, what it figured out is that it had to go through this tortuous trajectory and go to G in order to get this source of food. And it figured out that G is where the source of food is. Now, the experimenter flipped things around, added some surprise element to all of this. So you have the same sort of a circular chamber, but instead of this one opening out here, you have several different openings, and this one trajectory out here is actually blocked. So this particular opening is blocked. All right. So the question out here is, so this particular point is the equivalent of G. All right. So that's the physical location of where the reward was in the earlier setup. That's out here. Okay. So now the question is, how's the rat going to get to the reward? And there are multiple ways it can do this, right? One of the ways it could have done this is that if what it learned out here was, I'm going to take 5 steps straight ahead, I'm going to take 3 steps to the left, 2 to the right, and so on. If it was doing this step counting and figuring out the exact trajectory, then you know that, okay, since this one is blocked, it's probably going to go to the arm 10 or 9, something close to it, if that were the case. However, if the rat has a sort of map in its head, and upon this map it knows that the reward is somewhere out here, then what it would do would be that it would go straight through say 5 or something like that. It would take this trajectory right through 5. And it wouldn't be able to do this unless it had a representation of space in its head, unless it had a representation of the environment in its head. And that's exactly what the animal does, right? It takes this shortcut and goes straight to the reward rather than trying to explore what was initially a tortuous path. So it sort of figured out a way that this is the vector that I need to follow to the reward. Yes.
Smell the food, right?
No, so they've controlled a lot of these things, right? So they do make sure that it won't have any odor. Every time the animal takes the maze, they typically, and I know this from a lot of experiments, they'll clean the floors with alcohol. So they take a lot of care to figure these things out.
But then it's possible that the rat would go to any of these tubes and
we can also control for that by not putting the food there. We don't need to put.
No, you don't need to put.
Yeah.
All right. So let's ignore let's ignore Tolman's example. Imagine I bring you to the door out there and blindfold you, and tell you. You've sort of taken a look at the chairs and the tables around, and I tell you that can you navigate to the diagonally opposite corner of the room, and you'd be able to do this reasonably well even if you stumble upon various things, and that gives you a sense that there exists such a thing as a map in your head. So I could have perhaps used that instead of using this example. Nevertheless, I should say that it is quite controlled. They were very careful about a lot of these experiments. All right. So what this really implies is that there is a representation of space in your head. And for the longest time, people were looking for this, but they couldn't quite figure out where it was. And that was until John O'Keefe came into the picture. And John O'Keefe shared the 2014 Nobel Prize with Edward and Maybrit Moser for their discovery. So John O'Keefe, along with Dostoevsky, discovered place cells, right? So what are place cells? So he was, of course, looking for, or maybe he was rather primed to look for a representation of space based on his reading of Tolman's work. So here's the sort of experimental setup that you have in John O'Keefe's experiments. So you have a rat, and there is this enclosure, and then it's walking around, or sort of going around in this enclosure using some random trajectory, right? I do know that in a lot of experiments, the rats are thrown bits of chocolate-covered cereal, and that's what. So you just randomly throw it around, and the rats are going around picking this. So there are some benefits to being a rat, but here's nevertheless. So here. And yeah, they also stick a wire into its brain, so that chocolate doesn't compensate for that. But so all right, so you have a rat that's going around in this enclosure, and you have an electrode sticking into, in this particular case, into the hippocampus, in the CA1 region of the hippocampus, and it's measuring signals that are coming from your CA1 region. All the while, as the animal's going around, you also have a camera that's telling you where exactly the animal is in space, right? So now you have a way in which you can measure activity in the brain, and you have a way in which you can tell where the animal's located. Now, when you do that, here's an example. The gray trajectory out here is the trajectory of the animal going around, and this is probably of the order of 10 minutes of running around, if I remember this right. And the red dots that you see out here, those are the locations in space where a single neuron that they're recording from fired a spike, right? So these are the locations at which spikes occur. Now, here's the interesting thing, right? So when the animal's sort of wandering around out here, that neuron isn't firing, right? But when it gets to this circumscribed region of space, then the neuron starts to fire, right? And when it leaves that region of space, it stops firing. And it doesn't matter which direction it's coming from, it doesn't matter how fast it's coming into this particular region. The neuron's going to fire when the animal is at a circumscribed location in space. So these cells are what are known as place cells. All right. Now, for the longest time, people were, of course, wondering how do you get place cells, right? How can one come up with place cells? And there are several models to look at how one can make place cells in a neuronal network. But what, yeah.
So basically, observing some aspect of its environment,
Right.
which is sort of leaving a kind of signal or a sort of inducing a signal in that particular neuron. And there's also why it's not just in that area. There are also a few stray dots elsewhere,
Yeah.
which could also imply that it is getting some signal, some similar signal in the other region, but it's mostly in this one.
So what you're suggesting is that there is a stimulus and the neuron is responding to that.
Some stimulus from its environment.
So, you know, and this will be more true when we look at grid cells, but also as far as place cells are concerned, even when so there are some experiments where, you know, you train the animal to go around in a maze and so on, and then at some location in your maze, they put the animal on a wheel and it's actually running in place, right? And very often what will happen is that these place cells that fired when the animal was moving will also fire when the animal is in a particular location. And that, so it's called replay, for example. And very often in this particular experiment, they could predict with nearly 100% accuracy whether the animal was going to turn left or right. So it was actually very predictive. So it doesn't have to be, so the cause for this need not be the proximate cause for it need not be a stimulus that occurs. Right? So one should, the reason I bring this up is because one should perhaps not think of it as a sensory neuron with a receptive field. Right? So it is a few synapses further down from sensation, and there is a lot of different ways in which this can fire, and there are a lot of different circumstances in the absence of a stimulus where it can fire.
And another thing, this is this place is something relative to some origin, right? So if it's not confined to this sort of chamber, it's allowed to sort of walk freely. Then suppose then one does the recordings. The place cells would be firing with this. I mean, so it would measure some displacement with respect to some origin. The origin was also moving.
Right. So if you have if it's exploring freely foraging, you'll still have place cells. At the time that John O'Keefe was doing these experiments, it was very difficult to have larger enclosures. They were constrained to using smaller enclosures, also because of being able to record from a moving animal over long distances. So, for example, you'd need some track on top where this device would have to move along so that you're not tugging at the head and things like that. So there are a lot of different technical constraints that he faced, which are sort of taken care of now by a number of people who have even wireless recordings, are able to take care of a lot of these technical restrictions. And even in much larger environments where the animals are freely foraging, you can still see circumscribed patterns where the circumscribed regions where the neurons would fire. Yeah.
Find this particular thing. Did you randomly poke around and
Serendipity. Actually, so you should see the first paper that they published. It was just a few pages long, and it had recordings from, I think, 17 neurons from 17 neurons from CA1, and they fired at some location in space, and there wasn't a lot of statistics and p-values or nothing of that sort. It was yeah, these neurons fire here, let's call it a place cell, and very convincingly. So you didn't have to do things statistically for a lot of this, because it's not like 51% of people believe angels exist, therefore they do, kind of an argument, right? This is a damn place cell, this is where it fires. So it was really very prominent example. So this is what actually got a Nobel Prize. 17, the first recording was 17 neurons, of which 12 were place cells. All right. Did I answer your question? I forgot your question while I was going on a tangent.
Yeah.
Yes.
Did he just poke around and?
He poked around. It was serendipity. But not just serendipity. There was a sense that a hippocampus is one region where you should be poking around. The problem is also the hippocampus is really deep, right? You have to get there to CA1 is actually quite a bit of work. All right. So clearly here's a particular region that in the brain where location seems to be encoded. There are many nuances to it that I'm not talking about. There are a lot of things that have happened since John O'Keefe and Dostoevsky's initial work, but that's the broad sweep of it. All right. Now people started trying to figure out how do place cells come up, right? Can we look at the inputs that place cells give? Because this is rather perplexing, right? That you have neurons that fire at a particular location. So people wanted to look at inputs to place cells, and that, it turned out, was actually quite hard, because so there are multiple regions that give input to CA1 in the hippocampus, and they could sort of chemically ablate one of these regions, and that didn't seem to affect the place cells. The other region, which seemed to give connections, turned out to be very difficult to record from, because it's hidden deep in the temporal lobe. There's a blood vessel very close by. You have to navigate around it in order to get to this region. So these two people, this is Edward and May-Britt Moser, who are in Trondheim, and they shared the Nobel Prize with John O'Keefe, with the help of many friends, it turns out. Menno Witter, who's a spectacular anatomist currently out there, helped them navigate their way to the entorhinal cortex to get a sense of what's the input that place cells give, what's the input that place cells get from the entorhinal cortex. And here's what they found, right? So here you have inputs that are coming. So this is sort of a slice of the brain. The CA1 is where one of the places where you will see place cells. One of the inputs to CA1 comes from the entorhinal cortex. They were looking at the medial entorhinal cortex. There's the medial and the lateral entorhinal cortex. They were looking at the medial entorhinal cortex and inputs from there to place cells in the CA1 after much work. And what they came up with, what they saw when they recorded from neurons in the medial entorhinal cortex, is really this remarkable crystal of activity, as one could call it, right? So the gray lines, which are barely visible out here, is the trajectory of a rat that's running around in a square enclosure, going all over the place, and the black dots is the spikes, the firing of a single neuron in the medial entorhinal cortex as the animal explores space, right? And you can see what's happening out here. If you take any one of these blobs of activity, you will see that you can fit a rather nice hexagon around it. So you have a triangular lattice. So the neuron that you're looking at out here fires at the vertices of a triangular lattice that stretches across space. And these were really difficult recordings to make, one for various technical reasons, but the other actually related to the question you
This actually sort of answers my question.
yeah. So the other reason was very simply that when O'Keefe was doing his recordings, the enclosure was kind of small, and both Edward and Maybrit Moser were postdocs with John O'Keefe, and so they were doing the same size of enclosure while they were recording from grid cells. And what they found was that there was spatially tuned activity that you could see in the entorhinal cortex, but they couldn't quite make sense of it because there was one blob out here and one blob out here, and that would persist. Sometimes they'd see two blobs somewhere else and so on. So you couldn't make sense of the whole pattern. And I think it was a theorist that they were talking to that actually suggested, hey, maybe you need to have a bigger enclosure to see the whole pattern. And they made a bigger enclosure in order to do the recording, and lo and behold, that's what they got. Yeah.
The clusters are way less than the center.
The spikes on the boundary are very less. So, so while I say that this is a very nice crystalline triangular lattice, it turns out that the boundaries do affect the patterns, and we know of this from a little later recordings that came up a little later. But the fact that you have fewer spikes or fewer blobs at the boundary is just for this cell. I've seen cells that would have more blobs at the boundary. So that might be just a cell specific thing, right? Yeah.
Blob a neuron, or the entire thing is one neuron?
The entire thing is one neuron. So this is the way it's going. Imagine you have a floor that's tiled in triangles, all right? And I walk, every time I reach the vertex of a triangle, this neuron fires. When I leave it, it stops firing. When I get back to another one, it fires, right? So that's the crystalline structure that that one is looking at, right? Now, of course, you have a hexagonally close packed structure, and physicists jump in, right? I mean, there's no way to escape it. But okay, before we get there, so the properties of grid cells, right? It's quite remarkable. So so these are recordings from 3 different grid cells in the medial entorhinal cortex, and it's a circular enclosure. So you'll see that these the spikes occur at, they all of these spikes have this triangular symmetry, but if you look at the center of these grid fields, you will see that they are all shifted with respect to each other, right? So different grid cells have firing patterns that have the same symmetry, but they are shifted with respect to each other. And we now know that if you look at grid cells in the vicinity of each other in the medial entorhinal cortex, the grids are they're translated in phase, but they are never rotated. So there are no rotations out here. They're just translated, right? So all of the grids in the vicinity of a particular region in your entorhinal cortex will have the same angle of rotation, but the phases will be shifted all over the place. You can turn the lights off. So you have lights on. Then you have a period of maybe about 10-15 minutes where the lights are turned off, and you will see that the grids persist in darkness. Now, one could say that there are other stimuli that can perhaps anchor the grid and so on, and that may be true, but nevertheless light is not a constraint, and when you turn it on, it continues. The second is that if you look at how these grids are set up, right? So person walks into the room, looks around, and it seems like the grids are set up right away. So the first time the rat goes in, you see this sort of blob of activity out here, and over time you'll see that the spikes sort of coalesce around it. Coalesce meaning that particular bit of activity persists throughout, right? So it seems to suggest that something that happened in, the rat enters the room, neurons start spiking at the vertex of a grid-like tiling, and that neuron is going to continue spiking in that region, right? So it doesn't take very long for this grid-like pattern to be set up. It doesn't take visual stimulus to be set up. Yeah.
Defines the phase shift of each grids. Why do they shift?
So we'll try to address that in a model, but it's still conjecture. So we'll look at the network and try to find reason for that. But yeah, it is conjecture at this point. Okay. Oh.
So in this, they develop in a group. We can see that the center, the activation is
Right.
constant, or it's very fast.
Right.
Whereas over the time, it's developing regions.
So I see your point. So this trajectory was there, and maybe close to that, this group formed, but it's not out here. Yeah.
Is it like it develops in a blink, or is it like there is
Right.
center focus and then there is
Right. So it's it's difficult to say in it to that fine level of detail, but we do know with say place cells. So if you're looking at place fields and when does a place field pop up, it turns out that sometimes the animal might have to do 2 rounds, and in the 3rd round it will start forming, right? And there are a few theories about how that might happen, but no clear consensus on what is it that gives rise to it.
It can be said that it refines over time. If you take us in this room, representation of this room, it can happen in a blink.
Right.
But over time, the fine adjustments take place, and
Right.
we are more accurate about our position in it and navigate about it.
Right. Though I wouldn't say accurate out here, because the first spike occurs at some point, but then the blob actually gets bigger over time, right? I mean, it might be better, there might be better definition to it, but the size of it need not. And also, if one were to infer the location, you'd actually have to look at the population activity of all the grids. So that's a step further down. Yeah.
They are slightly, I thought they're slightly shifted.
Yes.
Because they want to cover the entire space.
Right, but you can't anthropomorphize neurons, right? And by that I mean you're giving it, when you say the neuron wants to do this, that's dangerous, right? Because I mean, sure, as far as the organism is concerned, it is important that the entire space is tiled so that any trajectory can be mapped to the sequential firing of neurons. But how that happens cannot be confounded with the fact that it tiles it is good for computation, right? So it's giving a human quality to a neuron. Yeah.
Can we also use conditioning to check the level of abstraction? So if we feed the rat consistently at one location in space, and then take it to a different location and introduce the food and check if the place cells associated with the previous place trigger at least slightly.
So that's a really good experiment, and actually people have done it. They've done it relatively recently. So in fact, one of the reasons they throw chocolate around randomly is really to because what people have known for a long time is that if you throw chocolate at one particular location and you're recording from a particular region, you will find a higher concentration of place cells in the vicinity of that. So there is a reward based change that is far less when you look at grid cells, but there are some changes that, yeah, sorry.
Assuming that these things could be erased out if they change the location.
Yes. So not necessarily erased out, they can remap to different extents. So here's an experiment that people would do. You have this circular environment. You put a post-it note at some angle relative to some segment that you choose, and you set up you figure out where the grids are. Then you take this post-it note off and you put it in a different place. So you just change the symmetry of things. And in grid cells, what you know is that there is a rotation in the grid to match with that particular change. So it does depend on the environment in this manner. So if you change the environment more dramatically, then you can have a more dramatic shift in the grid.
No, in the sense that if you change the environment,
Yeah.
you'll have to reuse some of these grid cells, right?
Mhm.
So you possibly have to.
Right. So if you change the environment, you would get a different grid field and all of that. But what remains invariant is the phase difference between any two grid cells that you measure from, right? So it seems to suggest that the topology of the network that gives rise to this pattern, that's what persists across, right? And that might be responsible for this grid. So yeah, in fact, even when you go off to sleep, these grids can be activated in sequences, and in those sequences, the phase relationship is still maintained. So it's a rather persistent relationship. The relative differences are the same. Yeah.
Maybe grid cells themselves have translational symmetry in space, in the
Right.
mind. And that is simply because of that they show this kind of translational symmetry in terms of I. Just structure.
That's exactly what we'll argue. That's what models argue, that the structure of the network that gives rise to these grids has a certain symmetry that allows these translations, right? And it also has a radial symmetry that gives rise to hexagons. But, yeah.
Also, these grid cells, through these studies, we can see that they are correlated to locations in space. But have there been studies checking whether they are essential to learning? So, if we eliminate them, can a rat still learn?
So there have been some. So, for example, bats also show grid-like patterns, and what one can do is one can try and eliminate grid-like activity in the bat entorhinal cortex, but that doesn't necessarily kill place cells. So place cell input can come from a number of different places. So there are a lot of different cells, but what we do know is that certainly if you take off grid cells or you disrupt the grid-like patterns, it seems to be strongly correlated with the ability of people to find themselves in space. So for example, in Alzheimer's, one of the earliest regions that get disrupted happen to be the entorhinal cortex, both the medial and the lateral entorhinal cortex. We know from some experiments actually done in MRI, and there are some properties of these grid cells that allow you to infer that there are grid cells in the medial entorhinal cortex. The symmetry of these grid-like patterns falls apart in people who have a genetic propensity for AD, right? So so it's actually quite interesting that it should show up even that early. So it does have a strong connection to navigation being the point. All right. So the question, of course, is how does one build a grid cell network? So now I should say that when you think about grid cells, right, it's these are not egocentric, right? It's not these patterns are not bound to where the animal is looking, how it's moving, and all of that. It's bound to the space around the animal. It's bound to the environment. So it's an allocentric representation of space, not an egocentric representation of space, right? The second thing is that because it's periodic, what you have is you can think of this like a scale with centimeter markings, right? So it's a way in which you can infer how much you've traveled. It's a coordinate system, right? So there the neuron fires, that's 1 centimeter, that's 2 centimeters, and so on. So you can actually, because of this spatial periodicity, you can think of it like a scale. But there's more to this, and I'm not sure if I put that slide in, but if you go from the dorsal to the ventral part of the medial entorhinal cortex and you record from grid cells as you go along, what you'll see is that the scale, which is the distance between these two adjacent vertices of your lattice, that's going to change as you go from the dorsal to the ventral side. But the change is not this continuous change. It changes in discrete jumps, right? And those discrete jumps, they follow a geometric progression. So it's something like n plus 1 by n. That's the ratio of the distances that you will see. So it just jumps in steps. And you can think of that as having centimeter markings, meter markings, and millimeter markings, right? So you can measure distances to various levels of accuracy, and you can think of the degree of accuracy as being the distance between each of these scales, right? So for example, if you're going really fast, you don't care what's happening at the millimeter level. You can just look at the larger scales. Turns out Uber has an algorithm that does pretty much that, right? When it's mapping out regions in space. Yeah.
If you say you have a grid field, right, and you are at the boundary of two grid cells, right?
Yeah.
Of two grid cells region. Now, if you are at the boundary, do both the grid cells fire?
Yes, so each of them has a firing field if there is an overlap. In fact, it doesn't have to be the boundary. The two fields can overlap and both of them will fire. So that's why to infer where you are, you need a population.
And that also gives you information about.
That gives you spectacular information. And when you have all of these scales put together and you look at the activity of the population, you can actually infer the location of the animal up to distances that are much greater than the lattice constant that you see out here. Yeah.
Regarding the scale, is it always consistent or is there more fine graining around places of greater?
So there is more fine graining, and the fine graining comes from the activity of the grid cells. So as it enters, goes into, and also for place cells, as it goes into towards the center of the field, the firing rate of these neurons increases. The phase relationship that these spikes have with respect to an oscillation called a theta oscillation will change, and that's called phase precession. So all of this can give you finer grained information within a particular grid field or a place field.
So all this is sort of triggered by locomotion. For example, if you just sort of
Yes.
move the mouse around, if you're going to play move it around, then you will see it.
So you
Actually do.
you do see it. There are some experiments of mouse in a car. There are VR, so you can do it in virtual reality. So all the human experiments are done in virtual reality. So you can do it without physical movement, but you're right, physical movement is a very important component out here. There are certain oscillations that you won't see without physical movement, and those play an important role in
Is there a visual information?
in this case, there is a visual context, but there are also experiments with blind rats. I wanted to say blinded rats, but then that would have been too much to say, but that's, yeah, let's call a spade a spade. Blinded rats also have grid cells, and they use whisking in order to find space. All right. Okay, so how do you build this kind of a hexagonal symmetric grid-like pattern of activity? How do you make this coordinate system? And there are a couple of different classes of models, and I'm going to focus on a specific class of models known as attractor models. There's another class of models known as interference models, which basically I won't talk about much, but you can look it up. It's models that say that the activity of neurons or these grid fields come up due to the interference of sort of oscillations in individual cells, right? I won't talk about it. It's a really nice and interesting way to think about grid cells and place cells. In fact, John O'Keefe was the first person to suggest an interference model for place cells, which he then extended for grid cells. But I'm going to talk more about attractor models, and I think a great deal of the inspiration for attractor models and generating hexagonal patterns or any symmetric or any periodic patterns in grid cells comes from the early work of Turing and pattern formation that he talked about in this paper, Chemical Basis of Morphogenesis. So this is from 1952. Right. So in Turing's rendition of pattern formation, you have two sort of morphogens, right? And there are inhibitors and there are activators, and you need both of these to interact together, and you will get sort of blobs of patterns based on diffusion and reaction in these morphogens. So the activity patterns that you see or the models that I'll talk about have a lot to do with how Turing thought about pattern formation and morphogenesis early on from the 50s onwards. All right. So if you wanted to build a grid cell network, there are two things that you have to do. One, you have to be able to create a hexagonal grid. Right. So the activity has to form this hexagonal grid-like pattern, and the second, this pattern has to be related to the movement of the atom. Right. So we'll think of this as two sort of related problems and see how we can make each of these work. Yeah.
So this is something I'm aware of this literature, but something that puzzled over why it did not even affect your models, etc. So when I look at this, I tend to see more of a sheared square grid than truly hexagonal grid. So
Right.
I'm not exactly sure why.
So
exactly.
yeah. So I think the early papers that Moser came up, that the Mosers wrote in 2005, they picked grid cells that were juicy and hexagonal, right? But the fact of the matter is that it's not a perfect hexagonal patterns. There are defects in the grid that you'll see. There is a shearing that you can come across.
Not even that way. I mean, when I look at this, I just see a square grid and it just looks sheared. It doesn't look.
Oh, you mean like a rhombus that you see? That's actually the. So if you no, so what you're seeing, so what I actually pick is really a triangle, right? So that if you have a triangular symmetry, then you can build, then you get hexagons. What you're looking at is something like this, right? So that's a rhombus. That's exactly right. In fact, if you were to take a quarter of this, so no, I can't do a pen out here. Let's see. Right, so if you were to take a rhombus like that, right? So it's many rhombi like that, and in fact, if you were to identify this and this, you will actually get a torus. And if you look at the grid, every the activity of a particular grid cell is just a point on that torus. So thinking of it as rhombi actually makes sense. It really is a point on the torus. I typically think of it as triangles, which is yeah. All right. So so these are the two things, right? So how do you connect the pattern to movement? So if you have a trajectory that runs through space, as the animal tramples over one of these grid fields, it's going to generate spikes, right? So you need to set up the model so that as you introduce velocity, as you introduce movement of the animal, the spikes have to be generated to match something like this, right? So how does one go about doing this? So the answer, of course, is attractor models. So what I'm going to do is I'm going to talk about this class of models, and they have largely been very simplified models of neurons, and I'm going to then move to some of our models which include the biophysics of neurons and lead to some interesting insights when we think about grid cells. All right, so here's a simple example of what a neuron can be thought of as just a simple on-off switch, something like your Hopfield model, which had switches connected to each other. Now, you guys have done the Hopfield model, right? So I can sort of gloss over this. So you have excitation, which would tend to make these guys point in the same direction, and inhibition, which would compel them to point in opposite directions. If you had all-to-all excitation with a few guys that were out of line, in the next time step, they would all end up in line if they were all-to-all excitatory connections. If you wanted to get an alternating pattern like this, then you'd have to connect these black guys to the gray guys with inhibition, and you'd need to have excitation in this pattern, right? So if you do that, then you will get an alternating pattern of ups and downs. You can embed any arbitrary pattern by sort of using that same connectivity rule, that if you want the same color, you have to connect them all by excitation, and if there are different colors, you have to connect it by inhibition, right? And if you do that, you will get something like this. You will get an arbitrary, you can embed an arbitrary pattern. If there's some noise in the pattern at time t, it'll go and fall into, so that's your associative memory, right? It's going to go and fall into this pattern that you're into this particular dimple in your energy landscape. All right, and of course, this is something that got Hopfield the Nobel Prize in physics in physics last year. So biologists are getting Nobel Prizes in physics. So there you go. I know people would argue that point, but we'll just appropriate them all. Okay, so this is a simple attractor model. Now, if you, rather than write a 2 out there, if you wanted a hexagon, you would just have to appropriately connect the network, right? So if you wanted a hexagonal pattern, what you'd need to do is this blob of greens would have to be connected by excitation, the surrounding blob would have to be connected by inhibition, and further out you don't need to have any connections, right? And if you had something like this, you'd get if it's a radially symmetric connectivity kernel, you'd force these to have a hexagonally closed packed kind of a structure, and you'd get a hexagonal pattern. But here's the assumption that one is making, right? So we are making this mapping out here from one space. This is a space which essentially consists of a sheet of neurons, right? So each of these points correspond to one neuron, and each neuron is either up or down, right? So that's in your brain, that's on your entorhinal cortex. This is measured in real space as the animals running around, right? So somehow we are saying that there is a mapping from brain space to real space, and we can do this mapping in a rather simple manner. Now, it turns out that this particular mapping from real space to neural space, there is or there seems to be some evidence that this is indeed the case, right? So people have done recordings from the medial entorhinal cortex in rats as they are navigating, and what they have found is that the shifts in the activity patterns, the coherent shifts in activity patterns, seem to match the direction of movement, right? So it seems to suggest that, one, there is probably some localization in connectivity in some manner, and two, that there is a mapping between these two spaces. But I should say that this is still conjecture. It's not maybe it's a little looser than conjecture, in fact, at this point. It's a wild guess that seems to be gathering some evidence in the literature. Yeah.
I know that I myself also aware of these attractor
Yeah.
models that we have discussed
Right.
a few times.
I was just wondering, has anyone tried also thinking these in terms of like neural field models? So different connection topologies.
Right.
Because the symmetry breaking in connections
Right.
may give rise to these.
Absolutely.
You know.
Yeah.
So I was just.
Yeah, definitely. In fact, I think if you look at most of the attractor models that you see out here, you can literally think of it as neural field
Correct.
models.
It's
So
a
you can have a
generalization from
continuum.
which you can
Exactly.
get this.
Yeah. So you can, instead of the summation in the attractor models, if you replace it with an
With some continuous
integration
function,
and a
yeah.
continuous function, you wouldn't lose
Right.
anything. In fact, people implicitly assume that it is continuous. Yeah. So you're right. It does match what you would see in a neural field model. All right. So this is one of the assumptions that we are making. Real space and neural space have this mapping between them. All right. Okay, so that's the first thing. And how does one go about building this attractor model? So let's look at how you do this on a line. Let's say you have a line of neurons, all right, and nearest neighbors are connected via excitation. In fact, I think a lot of attractor models don't require the excitation. They require relatively low or lack of inhibition is sufficient, right? But let's assume that you have something like a Mexican hat connectivity where you have excitation and you have off center or surround inhibition. So this is the connectivity kernel of this particular neuron, and the next neuron also has the same connectivity kernel, right? It's the same kernel that's just translated along this line. And if you have something like this, if you stimulate this whole line of neurons, you will get a firing rate profile that will give you this periodic kind of a structure, right? And that's basically the broad sense of what you'd see in reaction diffusion models. So when you do this in 2 dimensions, so you just have a radially symmetric connection pattern. So the neuron is at the center out here, and the connections to its nearest neighbors are excitatory. A little further out it's inhibitory, and then further out you have no connections. So this radial symmetry of the connections is what gives you this particular triangular or hexagonal lattice that you see, right? So you have a sheet of neurons and they're connected like this, and you're going to get a hexagonally symmetric pattern. And this is work from Burak and Fiete, and there have been a number of other models that also achieve some of the same goals. Yeah.
If we don't have excitation and only have lack of inhibition, then it is also possible that like some neurons from one cluster and some neurons from a very distant cluster fire together and start getting associated.
So here the neurons from this cluster and this cluster actually fire together, right? So there are distant clusters that are firing, right? All the blobs here are firing at the same time, okay? Now, the distance between these blobs is actually determined by the distance between these inhibitory lobes of your kernel. Now, how do you couple it to velocity? Well, so now you want this particular grid to, so there are 2 ways that you can think of it. I don't know if I put this. Okay, so there are 2 ways that you can think of modeling this system, right? Now, how do I connect it to movement? So either I can model the rat running along this trajectory, right, and crossing over the grid fields, or I can model the grid fields sort of going back on the rat, right? And so if instead of the rat moving down like this, if I had the grid field going up, it would really be the same thing, right? It's just a change of reference frame and you'd still get the same spikes, okay? So how do I model velocity? I can model velocity as the movement of the grid on the sheet of neurons in my brain, right? That's how one would model the movement of the rat, okay? Now, yeah.
You are suggesting that the neurons and phase shifting
Yes. Yes. So what I'm suggesting is that, so let's say you have a pattern, a hexagonal pattern of activity, right? When you move, that pattern of activity is moving with you, right? So that will give you the same effect, as it's basically just, rather than going into the frame of the person moving in space, you just jump onto a different frame of reference and then you're sorted.
In terms of looking at firing patterns.
In terms of looking at firing patterns, yes. All right. So now the problem out here is that you have this really symmetric kernel, right? And if you have this symmetric kernel, you're going to get a stationary pattern. Now, of course, because this kernel is translated all along this line, if I shift this pattern, it's still going to be a solution, right? It's going to be reasonable because the kernel is just, it's translationally invariant. So any translation of this particular periodic pattern is still a valid solution to this system. Right? Now, how do I make this pattern move in one direction and not the other? So one way to do that is that if I introduce an asymmetry in the kernel. All right. So if I introduce an asymmetry in the kernel, then I'll get a pattern that's going in this direction. If I switch the asymmetry and move it out here, I'll get a pattern that's going in the other direction. All right. So how do models actually achieve this? So you can think of it like this. So you have a line of neurons, okay, and there are going to be some neurons to the right of this neuron that have an asymmetry in one direction and some that have an asymmetry that is leftward, right? So you sort of randomly drop neurons which have connectivity kernels that are asymmetric in one direction or the other. Now, if you want the kernel, if you want the activity pattern to move leftward, you just select those, or you give input only to those neurons that have a leftward asymmetry. All right. And if you want the pattern to move rightward, you just turn on those neurons that have a rightward asymmetry. So just by changing the input to these neurons, you can move the pattern in one direction or the other. Is that reasonable? Yeah.
So we also have direction cells embedded into space.
Exactly. Yeah. So you do have cells that signal direction and so on. So the general idea would be that these direction cells are targeting specific groups of neurons that introduce an asymmetry in a particular direction. And that's something that you can also do in 2 dimensions. So if you have a connectivity kernel that is shifted in one of the 4 cardinal directions randomly, right? So you have a sheet of neurons and some neurons point north, some neurons east, west, and so on. If you choose the appropriate group of neurons to stimulate, then your pattern will move in the direction that's determined by which neurons you stimulate, right? So this is an example of simulations done by Govind, who was an undergrad in my lab. He modeled this, and you can see, right? So if I were to, if I think of it like this, if I'm recording the activity of neurons, so this is a sheet of neurons, and I stick an electrode out here, it's going to fire every time some blob just goes past it, right? And that's going to match the movement of the animal in a particular direction, right? So that's how one makes these models. Now the problem with these, I'm out of time, I'm guessing, right? What time? Some. All right. So the problem with these attractor models is that when you look at these equations that describe the attractor model, so this really tells you what the intrinsic dynamics of each of the nodes are. Right. So there are 2 components out here. That's the intrinsic dynamics of the nodes, and this term on the right is how they communicate with each other. Right. So the intrinsic dynamics of the nodes, you can see, is quite linear, right? You kick it, it's going to go up, and then it's going to decay exponentially. Right? That's all that each of these nodes can do. So all the complexity in the model comes from this particular term, where your f can be either a sigmoid or a some sort of a rectifying nonlinearity or something of that sort, right? But if you look at real neurons, this is not what they do, right? So if you look at recordings from stellate cells, they are remarkably complex. They have a huge dynamical repertoire. They have various ion channels. One of the key ion channels that we, channels that we look at is what's known as a hyperpolarization activated depolarizing current. So what that means is that when you inhibit the neuron, meaning when you hyperpolarize the neuron, that's when this channel opens. And the channel opens and it starts depolarizing the neuron. So even though you're inhibiting it, there is a channel that opens up and takes the membrane potential closer to threshold. Right? So that's this IH current, and you can see what happens when you hyperpolarize it to different extents. This particular curve that you see gives you characteristic time scales that are associated with some of these. If you look at the sub-threshold activity, when the neuron's not spiking, you'll see that it resonates, it can resonate at some frequency, and the frequency that it resonates at depends on the conductance and the time scales that are associated with various ion channels that we model out here. Now, it's also interesting that very often this resonance frequency is close to what's known as a theta oscillation. A theta oscillation is an oscillation that drives the medial entorhinal cortex and the hippocampus. It literally bathes the neurons in this oscillatory input that comes in from the medial septum, and the frequency of that oscillation is very close to the resonance frequency of individual neurons. So the question we asked was, how do you put all of this complexity in and yet get the grid-like patterns that we see, right? So it turned out to be a rather pesky problem as far as dealing with the parameters and trying to generate, do what simple neurons would do, right? And I'll tell you why it's worth doing that. So, all right, so if one were to make an attractor, how would we go about it? The first thing that you need to do is to take these biophysically detailed conductance-based neurons and construct switches, right? Because that's essentially what goes into an attractor model. And here's a switch. So here you have two inhibitory interneurons. You have a stellate cell. These are the grid cells. Inhibition drives the stellate cells, and that in turn drives the inhibitory interneuron. So what you see out here is a situation where when one of the inhibitory interneurons are firing, the other one can, you can switch it by just bumping it with an excitatory pulse, and you will get a switch taking place. So you get switches using, rather than using single neurons, you have to use network motifs, small motifs that will generate these switches. And now what you can do is you can connect these switches in a ring, all right? And when you connect these switches in a ring, you introduce asymmetries just as you did in the simple grid cell model. So there's an asymmetry from your stellate cell to the inhibitory interneuron. So the outer ring has an asymmetry going in one direction, in the counterclockwise direction, and the inner ring has an asymmetry going in the clockwise direction. All right. So depending on which of these stellate cells you stimulate, your activity pattern is going to go in the clockwise or the counterclockwise direction. And that's what we do out here. So if you give input to both the rings, your stellate cells are just going to remain stationary. If you give input to one of the rings, I think the outer ring in this case, you will get a pattern that goes in one direction. And so these are the stellate cells in the R1 ring. So it's in this ring. These are the stellate cells in the inner ring. And when you give input to the inner ring, you will see that the direction of movement is flipped, right? So what you can do using this is basically selectively stimulate one or the other of the other ring, and the pattern will move leftward or rightward. So we are able to create that using reasonably realistic networks, and the sizes of the grid fields, not surprisingly, are determined by the distance of the inhibitory connections. All right. So now if you wanted to extend this to 2 dimensions, right, instead of a ring, you would have a torus, and the remainder of the connectivity remains the same. So you have some east, sort of north, south, east and westward asymmetries. So that just tells you what the connectivity kernels look like on the torus. And when you do that, so here you can see the position of the animal, and I'm going to stimulate this each of these tori depending on the direction you want the animal to go in, and you can see the activity of different sheets of neurons, and you will see that as the animal is moving, right, you will see different sheets of neurons go up. So as it goes north, as it goes south, you will see the south sheet, west, east, and so on, right. So you can get this rather nice pattern of activity emerging even from realistic conductance based neurons. Now the question is really this, right? Why build a complex model when a simple one will do?
So the previous one,
Yeah.
how many neurons were there?
So this is each of these sheets are 50 by 50.
About 50.
So that's 2500 in each sheet. So this is 10,000 stellate cells and 2500 interneurons, and these are all single compartment conductance based neurons. All right. All right. So
That's also a lot of parameters.
yes, it is a lot of parameters, but a lot of these parameters are constrained. They're constrained by what the conductivity is of what these neurons do in experiments. All right. So the key question is, we started off with a really simple model that was making grids, that was doing everything accurately, and then we just went ahead and complicated matters, right? We put in conductance based neurons. We didn't really need it theoretically because things were all good. Why do you want to throw a spanner in the works with complicated neurons? And one answer to that is sort of attributed to George Mallory, which is because it's there. I sort of think it's a terrible answer. I think George Mallory didn't make it to the top of Mount Everest, but that's not the reason it's a bad answer. I mean, there are other better answers. One, perhaps Occam's razor, that you minimize the number of things you minimize the number of parameters, you take the most simple model there is rather than building complex models. There's another quote, and depending on what you think, one can find the right quote for it. So in Einstein's book on relativity, in the preface, he says that I adhered scrupulously to the precept of the brilliant theoretical physicist Boltzmann. According to him, matters of elegance ought to be left to the tailor and to the cobbler. As in, I don't care how ugly my model looks, I just want it to do the right thing, right? And I'm a Marxist in that sense. A Groucho Marxist, where I say that these are my principles, but if you don't like them, I have others. So depending on the circumstances, I'll make an idealized model or a simple one. But there are very good reasons, I think, to make a complex, detailed model. And here's one of the reasons which doesn't pop up when you look at idealized models. All right. So here's a set of experiments. This is very recent experiments, 24 and 25, where what you have is an animal that's running along a linear track or a maze or something like that. And you measure the activity of neurons in the entorhinal cortex as the animal's running around. And what these guys found is that if you look at grid cells, and you take the population activity of grid cells and try to infer where the animal is located based on the population activity, what they found is that the location that you end up inferring from the activity of neurons is not the current location, but it is where the animal will be a couple of a little distance away, right? So that's what these guys found. So the grid cell activity doesn't necessarily tell you where you are, it predicts where you're going to be. So that's the first thing. The second experiment, and I thought this was really interesting, is that they had this animal running along a trajectory, all right, and they measured the activity of grid cells, some class of grid cells, as the animals running down this trajectory. And at each point in time, what they found was that if you inferred the location of the animal from the activity of grid cells, they found that the inferred location would sweep leftward and rightward, right? So the animal was not trying to figure out exactly where it is, but it's trying to scope out the space in front of it, right? So there is a great deal of decision making that's associated with it. There is a predictive component to the activity of grid cells that comes up because you have to look at what's in front of you and you have to explore the space in front of you. So there are a lot of grid cells that are doing exactly that. They're sort of scanning the area in front of them. Now, this sort of activity is not something that one can capture using a simple model, because the simple models just tell you where you're currently located. It's not going to do this kind of predictive coding. So what we found is that if you have this particular channel that I was talking about, this hyperpolarization activated channel, when the animal is at a particular location, the presence of this channel actually ensures that the timing of spikes are taken forward, that it's hastened, right? So it makes it advances the timing of the grid cell spikes. And when you knock out this particular channel, as people have done in experiments, this is no one's actually seen this in an experiment, but we predict that if you knock out HCN, this hastening of the onset of the grid field is going to stop, and you won't see this particular predictive coding that we see out here. Without going into details of how this happens, the other thing that we found is that there are two ways in which you can generate a predictive code. One way is using this HCN channel because it hastens the onset of the next grid. The second is by introducing an asymmetry in the network. All right. And what recent experiments have shown is that if you look at grid cells in layer 2, those turn out to be the ones that hasten the advance over timescales that are comparable to what you would get from the HCN channel. Whereas if you go deeper into the entorhinal cortex, the distance that's predicted is much further down, and in order to have that, you would require asymmetries in the network. So we think that as far as entorhinal cortex grid cells go, based on our model, in layer 2 of the entorhinal cortex, it's the HCN channel that gives you a predictive code, and deeper in the entorhinal cortex, it's going to be network asymmetries that give rise to this. All right. So in conclusion, one, of course, we built a biophysically detailed grid cell attractor model, and this was not merely an exercise in self-flagellation. It actually has reasonable consequences, in the sense that we were able to show that a model like this can in fact generate a predictive code that tells you where the animal is going to be. There are a couple of things that also pop out from the model. One is that it corrects errors in dead reckoning, and we can talk about it offline at some point. And it also helps us understand, what would happen in certain pathological conditions. So I'm going to stop there, and I'm already way over time, I think. So thank you.